A group of scientists who called themselves the Death Battalion tested a chemical on themselves, then went to a party and thought they were dying. Decades later, a different group of scientists proved red wine was never good for your heart. Both of these events revolve around the exact same simple molecule. Yet they represent two entirely different timelines of scientific discovery, unfolding completely independently of each other. Contrasting a chemical accident that took 3 years to solve with a statistical illusion that survived for 30 years reveals the reality of how alcohol manipulates human biology. This animation shows exactly what happens inside your head when you take a drink. Ethanol is a tiny molecule that slips right through the blood-brain barrier, rather than acting on a single specific receptor like most drugs do. Once inside, it immediately enhances GABA, the main inhibitory neurotransmitter in your brain. This slows down your nervous system, producing the relaxation and impaired physical coordination associated with being intoxicated. At the exact same time, ethanol blocks NMDA glutamate receptors, your brain's primary excitatory system. Shutting this down directly impairs your ability to form memories, which causes blackouts at high doses. Alongside these suppressing effects, the molecule triggers a release of dopamine within the brain's reward circuitry, strongly reinforcing the desire to consume more of it. This simultaneous multi-system chemical attack produces the feeling of intoxication while embedding the biological hooks that make the substance addictive over repeated exposure. Your liver handles the physical clearing of the drug in a two-step process. In this diagram, an enzyme called ALDH attacks the ethanol, converting it into acetaldehyde, a highly toxic, cancer-causing intermediate compound. Almost immediately, a second enzyme called ALDH catches that toxic acetaldehyde and rapidly converts it into harmless acetic acid, clearing the poison from your system. But roughly a third to a half of people of East Asian descent carry a genetic variant that produces a much slower version of that second enzyme. This creates a bottleneck. The toxic acetaldehyde builds up rapidly in the blood. This buildup triggers rapid heartbeat, nausea, and the intense facial reddening known as the Asian flush. It is a directly observable physical reaction to toxic accumulation, and scientists eventually weaponized this exact biological mechanism. In 1945 Denmark, scientists Erik Jacobsen and Jens Hald were at the Medicinico Pharmaceutical Company, investigating disulfiram as a potential treatment for intestinal parasites. Their lab maintained an institutional culture of self-experimentation so intense that the most dedicated volunteers were nicknamed the Death Battalion. Testing the safety of the parasite compound, Jacobsen and Hald simply swallowed the disulfiram themselves before heading out to a social gathering. Days later at the party, the pair had a drink. They immediately experienced a terrifying physical reaction, pounding headaches, severe nausea, and vomiting so intense they thought they were severely ill. They eventually realized the disulfiram had irreversibly blocked their ALDH enzymes. By shutting down that second stage of liver metabolism, they had artificially induced a massive, unbearable version of the Asian flush. Because this reaction was an immediate, reproducible fact of human chemistry, it took the medical community only 3 years to investigate the accident and patent the compound as antabuse, an addiction medication still used to violently deter drinking today. While antabuse targets acute alcohol metabolism, chronic heavy drinking causes an entirely different kind of physical devastation. Continuous alcohol exposure actively blocks your gut from absorbing thiamine, or vitamin B1. Left untreated, this nutrient deficiency produces Wernicke encephalopathy, an acute condition characterized by severe confusion, abnormal eye movements, and a complete loss of physical balance. If not corrected immediately, it progresses into Korsakoff. This is a severe state of amnesia where the damaged brain unconsciously fabricates entirely false memories to fill in the blank spaces. This neurological damage and confabulation are permanent consequences of long-term use, entirely separate from the temporary effects of intoxication. While the heart chemistry of alcohol was established in the 1940s, a wildly different narrative emerged in 1992. Epidemiologist Serge Renaud coined the French paradox, proposing that moderate red wine consumption actively protected populations against heart disease. The public immediately embraced the idea. But this observational data contained a subtle mathematical flaw, known as the sick quitter effect.
For decades, observational studies routinely took former heavy drinkers, people who quit specifically because alcohol had already ruined their health, and grouped them in with people who had simply never touched alcohol at all. Dragging down the baseline health of the non-drinking group created an artificial mathematical illusion. It made the moderate drinkers appear healthier by comparison, rather than showing any genuine biological benefit from the alcohol itself. The specific biochemical claims dissolved too. In 2014, a massive 9-year Johns Hopkins study of elderly Italians proved that resveratrol, the supposed protective compound in red wine, had zero association with cardiovascular health or mortality. The researchers behind the French paradox did not manipulate their data. They observed a real pattern, but separating the true biological effect from the structural bias of the comparison groups required statistical bias correction tools that simply were not applied until decades later. Correcting that statistical illusion brings the story right back to the chemistry discovered by the Death Battalion in 1945. Acetaldehyde is the exact same genotoxic molecule that made Jacobson and Halt violently ill. Because acetaldehyde actively damages DNA during the metabolic process, international health authorities classify alcohol as a Group 1 carcinogen, placing it in the exact same proven danger category as asbestos and tobacco. This hard biochemistry ultimately dismantled the epidemiological myth. In 2023, the World Health Organization issued a clear statement concluding that no level of alcohol consumption is safe for human health. Decades of genuine public health messaging built around the protective benefits of moderate drinking finally collapsed under the weight of real biochemical evidence. It took scientists only 3 years to turn a chemical accident into a medical treatment, but it took the broader medical community 30 years to untangle a statistical error about the exact same substance. To understand the full timeline of exactly how this molecule interacts with the human body, and for more deep dives into the science that shapes our world, be sure to join us for January Science. Keep questioning the data, keep exploring the chemistry, and we'll see you